that an antibody would not be able to bind to. B cells respond to surface. T cells respond to internal amino acids or internal sugar structures, internal to that protein on the surface of the B cell, the T cell, or on the surface of the bacteria, the virus, or the parasite. If the T cell can bind to the MHC, once bound, it can then bind to whatever this epitope is. Secondary, tertiary signals from the T cell will then go to that cell saying for it to die. Because if this protein, this epitope, is presented to the T cells will then respond such that it believes that this cell that's presenting this epitope, which is from the internal three-dimensional structure of a viral protein, is infected with a virus, and thus this cell needs to die. Better to lose one cell than lose a clump of cells, lose a clump of cells, lose the tissue, lose the organ. Organism dies. Now, I've mentioned a few times already the lymphatic systems, lymphatic nodes, the lymph ducts. What we have here is a secondary circulatory system. Our primary circulatory system, composing of the heart, our arteries, and our veins, delivers blood, oxygen, nutrients from our central core out to the rest of the body. The part of also what it's delivering is fluids. Fluids will leak out of the arteries, the veins, into the surrounding tissues, and if there was no way to drain this fluid, then we would swell up. As a side note, that's what elephantitis actually is. Clogging or abnormally formed lymphatic system means that the fluids that are being delivered by the circulatory system are incapable of draining out of a leg, and hence that leg will continue to swell up. Now, as the fluid's being delivered, it leaks out and it goes into the tissues. Once it's in the tissues, it'll begin to drain via the lymphatics. The lymphatics will take it back. It'll deliver it to the abdominal cavity or back up to the circulatory system to replenish the fluid levels. As it is draining out of here, that shows this leg. The bloodstream is delivered fluids as well as white blood cells, macrophages, dendritic cells, to the area of infection here on the foot. The fluid's draining. As it's draining, it's collecting into the lymphatic ducts. It's draining to the nearest lymph node, drains from that lymph node to the ones here, to here, and all the way through. As it's draining, it's also bringing along viral or bacterial proteins, polysaccharides, remember those PAMPs. It's draining those out because it turns out one of the things about viruses and bacteria is they are very messy. They are constantly shedding. One virus goes in, thousands come out of a cell, it causes the cell to burst, and there's all parts of little viruses that hadn't completely formed yet. Bacteria are constantly shedding the surface proteins, and they're constantly remaking their cell walls. So all of that will drain to the lymphatics. It'll tell the cells there that there is an infection. So what you have as you are going to the lymphatics, the lymph node here, you'll have what is referred to as a high endothelial venule. This is kind of like a large tube where smaller lymphatic tubes drain into one larger and then larger, and they come together and together, kind of like the drainage system you would have in your house. Once it hits, though, the lymph node, you're going to see that the lymph node itself has regions, follicles, cortex, T cell zone. Each of these different areas are going to have a different composition of cells present. So what you have here is the afferent lymphatic vessel. Fluid flows in from here, out here. this bean-shaped thing here is the lymphatic node, the lymph node. Fluid flows in. First thing it comes into contact with will be the primary lymphoid follicle. This is going to be composed mostly of B cells. These yellow regions you see here are what are referred to as germinal centers. These are going to be where a B cell has been activated. It has now gone from immature to a mature effector cell, or it was a quiescent, a resting mature cell that is now once again being reactivated and has gone through cycles and cycles of clonal expansion. Remember that mitosis. As the fluid flows through, that's going to either have bacterial or viral proteins in it. It's going to flow from the B cell to the T cell region here, this paracortical area. The paracortical area, again, comprise mostly of T cells. T cells get activated. They will flow out to the arteries and veins and then go to the rest of the body. B cells, T cells, finally here, what is referred to as the medullary cords. This is where macrophages are going to be sitting. So when in doubt, the last thing it's going to hit is the macrophage. The macrophage is not antigen specific. Anything not you, it will go swim out, follow the chemokine breadcrumb trail, try to find where the site of the infection is, and start phagocytizing to bring in new antigens to try to activate B cell, T cells. Fluid flows through along with the antigens. If the antigens can't bind anything in these three regions, collects, flows out, flows to the next lymph node, and to the next, and to the next. Get it? Now, we have areas within our body that are quite rich in lymphatic tissue. It may not be lymph nodes themselves, but it's lymphatic tissues in that it houses immature as well as mature B cells, T cells. Many of these areas, such as the periarterial lymphoid sheath, it's a T cell containing area that is looking for anything flowing by in the arteries. Some of this PALS tissue will be in the spleen itself. It's going to be spread throughout the body. It's going to have a marginal zone of B cells. So it's primarily T cells with some B cells there. B cells, remember, are going to be activated to mature into plasma cells. Plasma cells making antibodies. T cells are there to go hunt down, kill, as well as to tone down after the infection is gone. We also have what is referred to as a mucosal immune system, MALT. This is going to be things like your tonsils. It's going to be your adenoids, places like that. Starts at your sinuses, starts at your mouth, runs entirely through your gastrointestinal system. So that anything that's present there that may come into contact with the mucosal surface, anything that may be able to start penetrating through, will immediately run into a lymphatic tissue, mucosal associated lymphatic tissue. You also have gut associated. This is going to be more the intestinal, large intestine. And you're also going to have NALT and BALT, which are different areas, nasal, and I'm drawing a blank on what BALT is right now, but that's okay. Just know that these are lymphoidal tissues, associated lymphoidal, mucosal, gut, nasal. The spleen and many of these other lymphoidal tissues are not going to have extremely defined areas like the lymph nodes. These are larger organs. So their larger organs are going to have smaller areas. All of this is going to be cells, tissues that are there for screening and then maintaining the B cells and the T cells, or for replenishing red blood cells, removing dying or damaged red blood cells. When you look at a close-up of the spleen or some of these lymphatic tissues, you're going to see that it's a highly vascularized area. As the blood flows through, these fluid will flow out. With the fluid will flow out potentially antigens. The antigens will be picked up by APCs, which will then deliver to B cell, T cell rich areas. Blood flows on, in and out. As it flows through, fluid comes out, and away it goes. Here they're showing you a tissue stained slide so you can see the different zones. The different zones are going to be due to different cellular compositions of differentially differentiated cells. I know that was a lot to say, and that's quite a big mouthful, but as blood flows through, fluids will pass out with the antigens, and it'll flow through these different populations of cells. Now, it's not a distinct, here's this ring, and this is here, and that's there. They will mix through the different zones. The cells will mix, but they do try to congregate, as you're seeing here, these differentially stained than these. It makes it easier for the cells, the organs, because this macrophage in this area here, the PFZ, the MZ, the CO, these different areas, the PALs, are going to need different secondary cells to maintain the macrophages, the neutrophils, the T cells, the B cells. Now, when you look at those associated lymphatic tissues, the MALT, the GALT, what you see is that below the surface of the mucosal epithelial is going to be what are referred to as domes, subepithelial domes. Columnar epithelial here, making up the epithelial mucosal epithelium. Here is an M cell, M for, I can't remember. I believe it's named after somebody, but we're not going to worry about that. M cells, what this does is it phagocytizes. Phagocytizes something, pulls it across, delivers it here into the dome, where once in the dome, whatever it has phagocytized and digested, parts of it we presented here, dendritic cells will pick it up. Dendritic cells will pick it up, and then it will deliver to B cells, T cells. If it can't find anything here that recognize it, it will swim through the efferent lymphatics to the nearest lymph node and present there. What we're looking at here is an artist's rendition of what is referred to as a Peyer's patch. You'll find these all throughout the stomach and the small and large intestine. You can also find versions of this in the mouth, the sinuses, wherever you are going to be in contact with the outer environment. And yes, you have to remember, even though you've put something in your mouth and swallowed it, it is technically still outside of your body. Once it crosses this epithelial lining into the underlying tissues, only then is it actually inside of you. Now, why go through all this trouble? Why have the dendritic cells and all these other cells in the domes and the spleen? Well, because remember, it goes back to what I talked about in lecture 1.1. There has to be a co-stimulation, a co-switch. Yes, T cell receptor binding, interacting with something the dendritic cell is presenting to it. That is the first signal. That is the major signal, the major switch. But it also needs a secondary one. The secondary one is the final push that this T cell will need to go through and activate a whole section of gene transcription translation. This will then activate different cellular responses depending upon what this second signal is. Only so many MHCs, only so many T cell receptors. They bind here. This is where regulation and selectivity, how the T cell will respond, comes into play. We see the same thing here. What was bound to the T cell receptor is now going to be presented to a B cell, because this is an effector T cell. This is the T cell receptor presents B cell receptor, which is also known as an immunoglobulin domain, gets the first signal. Second signal coming through from the T cell, or from something binding to a different receptor. By having these different switches that all have to be on at the same time, different levels of regulation, different levels of finesse. There, one of these cells is going to present it to them. It's going to present it to them as the primary signal, and then also deliver a secondary signal to activate. Now, once the job is done, whatever was infecting you, bacteria, virus, parasite, has been removed. Remember, one of our regulatory T cells is going to tell most of the plasma cells to die. You don't need to make all of that antibody now. Not all of them are going to die, just the majority of them. Keep some around, keep them alive, so that way you have memory. Same with the T cells that got activated because it was a viral infection. Not all of them are going to die off. The majority of them will, but there will still be some around that recognize whatever that antigen was, that epitope. This is the basis of immunization. Some point in time, you got exposed. Either somebody sneezed on you, or when you were a little baby, you got a shot. After you got that first shot, you had what is referred to as a lag time. About a week or so that it takes initially for your immune system to gear up. This is why infants get sick a lot. Their immune system is not primed. It has to be primed. As it learns, it's being activated. You'll see right about that 1 week time, antibody response will start to go up. Serum levels of an antibody to whatever the epitope or the antigen was that was in the vaccine, or was in the virus or bacteria that they got sneezed on. It'll peak, it'll be maintained for the next 2-3 weeks, and then once the immune system has cleared out the infection and the regulatory T cells start to tell everything to calm down, it'll go down to a certain level. Never goes back down to nothing. Always stays above. Fast forward months, weeks, months, years later, there's going to be a second exposure. Say this was measles, the MMR vaccine for the 2-month-old. Goes through this, goes down. Then when the kid's 10, runs into somebody from an anti-vaxxer family who decided not to do it, and they got the measles, they get re-exposed. Well, instead of taking a week, it's now within a few days, a day or so. The immune response kicks in, and look how much it kicks in. It goes from less than 10 to the 1th up to 10 to the 4. Remember, this is log base 10, so that is 10,000-fold higher than it did at the primary, and it goes up well beyond. This is high. It'll maintain as long as there's an infection. Then it'll drop back down. Next time they're exposed because somebody's got the measles, or at least is carrying the measles, goes back up. Does so within a day. Doesn't take 1 to 2 weeks for the immune system to learn. This is immunological memory at work. Now, all of this works together. The cells, the B cells, the antibodies, complement that we'll talk about soon. All of these work together to go after specific types of pathogens. If you look here, bacteria, parasites, fungi, extracellular, intercellular, viruses, parasitic worms, we have been exposed to these since we mammals first crawled out of the mud holes. Over the past millions and millions of years, have these parasites, pathogens have evolved, so have we. It's kind of an arms race. And that's what we're going to be looking at throughout the rest of this course. These different pathogens, what did we do? How did we compensate?